Yeah. Because we're all.
I've experienced these.
Yeah, but,
That's what holding is those.
Yeah.
Alright, so I think we're gonna get started here. So my name is Tom Schmelk. I'm one of the forest entomologists for the Maine Forest Service. I'm based out of Augusta, but Winter Moth is one of my programs. So I would say this is probably my first pure brown-tail moth talk. Typically I'm talking about brown-tail moth or some of the other forest insects, but which includes winter moth, but never full winter moth. So this is a all brand new presentation, and hopefully there's no kinks. So going through it a few times. Okay, so I'm going to talk about winter moths biology, a little bit of its life history. Then I'll talk about the biocontrol agent that we have for winter moth, its biology and life history, and then I'll talk about current situation in Maine, and then management and mitigation strategies that homeowners can use. So it's a little blurry.
That's the zoom, and then if you twist the front of the lens there.
There you go.
Oh,
I thought it was really nice.
So winter moth is non-native. It's native to Western Europe, and over in its native range, it serves a really important role in two respects. Its first role, and illustrated by that sparrow on the side. So in Europe, a lot of the birds migrate down to North Africa and to the Mediterranean, and when they return in the spring, winter moth is probably one of the first caterpillars that's available in the spring for these birds to sort of refuel after flying all that distance. Then also as a food source to feed their young, which is what the sparrow is doing, is collecting these winter moth caterpillars in order to feed its young. And we do have plenty of birds that eat winter moth, although since it's not native, there's not really a co-evolution. So sometimes it takes a little while for these birds to key in on that fairly new food source. So winter moth's only been in Maine since about 2012. So these birds don't really have that co-evolution as the birds in Europe do. So most of you have probably seen the caterpillars, but they're these very small, or sort of about inch and a half long caterpillars with these faint yellow longitudinal stripes all the way down the body. So they're in the family Geometridae, which encompasses a lot of species of moths. But one thing that all geometrid caterpillars have in common is that inchworm type of locomotion, where they arch up. So these it's actually 6 legs. These 6 legs in the front, those are the true legs, and that's true of any caterpillar. And then the legs in the back are called prolegs, and they're not true legs. All insects have 6 legs at some point in their life cycle. So you got the 6 legs up here. So on the back of the caterpillar, those are just really extensions of the exoskeleton that help it grip. And in the case of inchworms or geometric caterpillars, it helps them move along the branches and the leaves. So winter moth has sort of a weird life history, especially compared to a lot of our native moths. So the females are apterous or nearly wingless. You can see the pointer, but you can see the female on the bottom has very reduced wings, and they can be even more reduced than that. And that is an evolutionary strategy, basically, so the female can put more energy towards egg production and doesn't have to waste energy growing wings and then using those wings and flying around. So how do these, this adult male moth, which is fully winged, fully capable of flying, so the ones that you see flying around in November, December, how do those two get together? So the females, since they're flightless, they will call the males to them with these pheromones. So they release a pheromone that calls these flighted males to them, where mating occurs, and then shortly thereafter, the female will lay eggs and the males will die after mating. So like I mentioned, winter moth adults are active in, it's typically mid-November through December and sometimes into January. Really depends on the weather. So if you can, or I have it on a future slide, but the adult females, so they're all pupa in the soil, these little tiny dirt pods. So pupa is that stage between caterpillar and adult moth. So the adults emerge out of the pupa in the soil, and since the females are wingless, they need to crawl up the trunks of the trees in order to lay their eggs. And they're trying to lay their eggs in bark crevices and sort of divots in the bark, but they'll also try to lay their eggs right on the bud tips, so that when the eggs hatch, the food is right there and the caterpillars don't have to climb very far. So that adult emergence in November, December, January, that's all very dependent on the soil temperature. And if we have sort of an early freeze and the ground stays frozen, that means those winter moth can't emerge from the ground, which is great for us. But we're not trending that way, unfortunately. But yeah, so fast forward, females are mating, they lay their eggs in the fall, early winter, and then those eggs sit in the bark or on the branches until the following April, typically around mid-April, once those eggs are hatching. And then the caterpillars will go and come out to the nearest bud, and that's mid-April is typically when the buds are swelling and the new leaves are emerging, and that's the perfect food for these small caterpillars. So emerge mid-April, and then they're going to feed all the way until late May, even the first week of June, but it's typically the last week in May they're completely done feeding. But they can cause a lot of damage from mid-April through May, as many of you have seen here. And the damage that I'll wait till the next slide. But the damage is sort of this shot hole pattern. Sort of looks like somebody took a shotgun to the leaves. We'll talk about that in the next slide. So also when the caterpillars are out, when they're a little bit more grown, one of the ways that they can spread is by dropping out of the tree on a silk thread, and that's called ballooning. So they're hanging on these threads, the wind comes, blows them like a balloon onto other foliage that's in the understory or into other trees. So that's one of the ways that they move around. And that's also a strategy to avoid predation, because once a tree's leaves are damaged, it releases these volatiles, and there's parasitic wasps, parasitic flies that are attracted to those volatiles because it's telling them that hey, there's caterpillars feeding here, and that's a good place to find something to lay their eggs in. So the caterpillars want to sort of distance themselves from that damage. So this is that shot hole pattern that I was talking about. So very heavy infestations like you have here on West Rhode Island and a lot of the other peninsulas in mid-coast Maine. They will consume the entire leaf right up to the midrib of the leaves. So all that photosynthetic material will be gone. They're not very picky eaters, but some of the most common things that we see them on in Maine is oak, maple, birch, any fruit trees, and then basswood and blueberry, and many others, but those are probably the most preferred hosts that we see. One thing to note, so a lot of people see this type of damage and with this or brown tail moth or in combination, and they see the tree has no leaves on them, they think the tree is dead. But thankfully a lot of oaks and maples in particular, they can survive and withstand a few years of defoliation, even in a row, and still be just fine. The only things that complicate that are drought, sort of like we are having this year, unfortunately. And then also other stressors like something else eating them, like brown tail moth, which will consume even the midrib of the leaves. So yeah, typically trees can last a long time and they're sort of pre-programmed to be defoliated and still rebound from that. With a few exceptions, as I mentioned, those additional stressors which can sort of push them over the edge. So these are those pupa that I was talking about. They're basically the size of a tic tac. And inside, basically, there's a pupa surrounded by a silken cocoon that has all this dirt that sticks to it, and it helps give some protection. So just a little bit about the history of winter moth, just in North America. So it was first introduced in Nova Scotia in the 1930s, and that population, hasn't really spread as far as more recent introductions. So that introduction in Nova Scotia has been here for well over 100 years. And there is some evidence that it has come into Maine, but nowhere near some of the more recent introductions. Came to Oregon in the 1950s, British Columbia around the 1970s, Southern New England in the 90s, and it's been in Maine since about 2012, maybe even a little bit before that. So one of the reasons why we have these repeated introductions, that I should also mention that each of these introductions is from a genetically distinct population in Western Europe. So just multiple introductions, a lot of plant material moving around, a lot of soil associated with that plant material moving around. And that's the major way that winter moth moves, and that's how it came to Maine. So I'll also talk about spread a little bit later, but yeah, just be cautious when you're moving plants from one place to the other, because winter moth can come, but also there's a lot of fungi and diseases and other insects that can move with that. So this is a little bit outdated map, but sort of gives you an idea of the range in Maine. So all the way down to Kittery, up through MDI, Washington County is pretty, doesn't have a whole lot of winter moth population, even in the blueberry fields and all that are up there. And even up to Lubec, there's just a very small population. Most of the population is where we are, right in the mid-coast. Down in Kittery, which has been quiet this past year. And then we have a growing population on Deer Isle. That's now, that defoliation is now visible from the air. It wasn't a couple of years ago. So that population is building, but the worst is this area of Mid-Coast Maine. And the worst towns here in Westport Island, Bristol, the whole Bristol Peninsula, Edgecombe, and Georgetown, Pittsburgh were all really, really bad. But there is hope. So
That's what we want to hear.
yes, usually these talks are sort of depressing, but winter moth is actually a good news story. So we have a fire control agent. It's a fly, true fly. It doesn't have a common name, which is kind of surprising. It's a very important insect in environmentally. So it's part of a family called Tachinidae, and that family is a very large family. It's probably one of the largest fly families, just behind a couple other families. So there's 8,200 species that are described worldwide, and about 1,300 species just here in North America. So all of those, all the flies in that family, all 8,300 species, they're all parasitoids of other arthropods. So when you think of parasitoid, the difference between a parasite and a parasitoid is that a parasitoid kills its host, or typically kills its host as part of its life cycle. So the larvae, typically the larvae of these flies, live inside or on the host, and the ones that are inside, they feed on the host. They typically are sort of pre-programmed to only eat the non-vital organs to be able to keep the host alive for
It's crazy.
for long, and then as they mature, in the host, then they will, consume the more vital organs. So it's kind of gruesome, but yeah, the difference between parasite like a tick and a parasitoid like a tachinid fly, pretty marked there. Yeah, so for most tachinid flies, the eggs are very close to, so it's not like a squash bug that lays a egg on a squash plant and it takes a little while for that egg to develop and hatch. The female tachinid flies, they sort of hold the eggs within their bodies, and there's a bunch of different modes of birth, I would call it. So some are almost live birth, so the eggs hatch within the abdomen of the female, and a maggot is laid either on or in the host, but most lay eggs. But again, they sort of keep them inside their body so that they're closer to hatching when they're laid. Because if you think about it, they're trying to lay eggs on or inside a host, and the ones that lay them on a host, there's the longer that egg is on the host, the more chance there is for something to happen to that egg on the outside. But flies like Cyzanthus albicans, they don't lay it on or in the host. What they do is they lay a lot of eggs on the host foliage that winter moth is feeding on. So I'll go back to the previous slide, but they lay microtype eggs. So microtype eggs range in size from 0.02 millimeters all the way up to 0.2 millimeters. And just for reference, that is the thickness of a piece of paper, or the particle size of very fine sand. So very, very small eggs. And they need to be small in order for that caterpillar to ingest them. But yeah, they're sort of the eggs, the type of microtype eggs that Cyzanthus lays, sorry. They're sort of ovoid, so they're twice as long as they are wide. Yeah, so let me go back here. So this word here, cynovagenic. So that's a really fancy term, meaning that instead of laying one batch of eggs, like a lot of insects do, and then completing their life cycle, cynovagenic flies, the females can keep laying eggs throughout their life cycle. So they continue to produce eggs and lay those eggs. So that one female fly can lay a lot of eggs in her lifetime, which is exactly what we need for something like winter moth that has a really high population. I should also mention that they are very specific to winter moth. So there is a closely related moth that's native to this area. It's called Bruce spanworm. It's in the same genus. It's virtually identical to winter moth. The only way to really reliably tell them apart is through dissection. But they don't really go after our native Bruce spanworm, because there's sort of a Bruce spanworm's active maybe a little bit earlier in the spring, and active a little bit later in the fall. So it's sort of it's not a preferred host for Cyzanthus.
Is this from Eastern Europe as well, or is this a native?
Cyzanthus is native to winter moth's range, so not native to North America. So with a lot of these biocontrol agents, they have to go through like typically a decade of really vigorous research, and they're subjected to all of the closely related species that are native here, to make sure that they can't survive on those hosts or don't prefer those hosts. So it's not like it was back in the day where they would go to the native range and pick up whatever was attacking that species that they wanted gone, and brought it over. A lot of those were generalists. A lot of those attacked our native species, but Cyzanthus is very specific, and so that's one of the reasons why it makes a perfect biocontrol. Okay. So microtype eggs produce microtype larvae. So there's there's certain things that, so eggs that are laid on a caterpillar, those larvae need to be able to penetrate the outer body wall of that host. So microtype larvae from those microtype eggs, those are typically ingested, so it doesn't need to break through the outer body wall. So they're smaller, they don't have the mouth parts, the sharp mouth parts that a lot of those other tachinid flies that are laid on the outside have. And like a lot of human parasites and parasites that live inside the body, they're very small, colorless, because they don't need melanin or anything like that to resist the UV effects of the sun, or camouflage or anything like that. Sorry, this is a little bit in the weeds, but a lot of insects have an open, have a tracheal system. So if you look on the sides of the caterpillar, if you were to look, say the tomato hornworms that are in your garden, if you look at the sides of those caterpillars, you'll see little dots, and those are called spiracles, and that's the opening to the respiratory system of the insect that goes into trachea and sort of oxygenates the hemolymph or the blood that's inside the body cavity. So with these larvae that live inside their host, they don't really have those need for those spiracles. They are able to diffuse oxygen from the blood of the host or the hemolymph of the host, and take that in. So they don't, they're sort of like scuba diving pretty much, except they don't need a tank. They can just take the oxygen from the water or from the hemolymph. Okay, so like I mentioned, we've been releasing this fly in Maine since about 2013. In about 2011, 2012, that's when we first started seeing problems with winter moth down in Kittery, but then also up near Harpswell. So we are very limited in the amount of biocontrol that we get each year. So the reason for that is in southern New England, winter moth was, running rampant, and the researchers at UMass Amherst were able to develop this biocontrol agent, or at least get some biocontrol agent from Oregon, which winter moth had been there since the 50s, and this fly had already been released out there to good effect. They brought the fly from Oregon back over to New England and started rearing them, and came up with protocols for collecting caterpillars in order to get more of these flies. So there's a great biocontrol program down there, and they released a ton of it in southern New England. And it was so successful pretty much that they can't really find large winter moth populations to collect from down in southern New England. So that's where we used to get our biocontrol, and they used to send us about 2,000 fly pupa each year, so that we could release it in Maine. So since we are not able to rely on that steady stream from southern New England, we basically have to rely on our previous release sites here in Maine, which we are having great success from, even over next door, Booth Bay Harbor. Released there probably 4 or 5 years ago, and you sort of see the difference. The leaves used to look like they did here, just sort of stripped, but going back to collect, each subsequent year, you just see less and less holes until pretty much you can't collect enough winter moth.
Did you say you get about 2,000 of these pupa a year, and that's enough to?
Yeah, I'll go into the numbers a little bit later, but there's a ratio of caterpillars to flies, and it's not a good ratio, unfortunately. So yeah, so basically we have to rely on just our previous release sites here in Maine, which there are many. We've been releasing pretty much once a year since 2013, so we have a bunch of sites built up.
How far do they fly?
Saisonets?
How far do they fly?
Saisonets?
Yes.
Yeah, so they will, so when I get to one of the other really site slides, so there's Kittery was probably one of our first releases. So 5 miles away from that, there's a nature preserve called Brave Boat Harbor, and that we have never done a release at. And we sort of got feed into that from one of the land trusts. And that's one of our most productive sites. So they can fly miles. I'm not sure how many miles I don't know if it's been studied how many miles they move a year. But it is miles.
I just wonder if we would get any in Westport,
Yeah.
from Georgetown.
You definitely would. So we have releases on both sides, but I have a list of the towns that we've done releases. So in order to get these caterpillars, there is a certain protocol that was developed by UMass Amherst and us to sort of streamline the process. So we will go back to the general area or even the specific release site that we've released these flies from before. Typically an area that has a lot of oak, a lot of red maple, and has low branches that we will be able to collect caterpillars from. So what we do is we spread out these tarps on, well, preferably a flat area, but there's some rocky terrain that we don't always have great luck in. And then we have these large poles, and they're sort of these extendable poles that are used for painting like cathedral ceilings. And we wrap the branches that have that Swiss gold cheese, that shotgun defoliation pattern. We wrap the branches, and it's sort of like, it's kind of like rice grain. You hear just rice grains, and it's all these little fat caterpillars that are hitting the tarp, but there's also leaves and branch material. And then after, we all get down on our hands and knees and pick up these caterpillars one by one.
Oh my god.
And sometimes we have these luxurious yogurt cups that we are able to put them in, so we don't have to hold them in our hand. But we have these 5-gallon pails that we put some peat moss in the bottom, and then we stick damaged leaf material in, and then we put about 500 caterpillars per a bucket in there, and we label it. We have these little clickers that when somebody's adding 20 to the bucket, so we don't have to keep count as well. So yeah, there's a lot that goes into that collection. So there was great need because, like I said, many of these peninsulas have really horrible winter moth populations with severe defoliation. Coincidentally, Rhode Island, so I mentioned the success down in southern New England, Rhode Island hasn't really shared in that success for some reason that all of the other southern New England states have. So winter moth is sort of rearing its ugly head there, even though there has been releases there of Sidenus in the past. And there's a lot of rich people down there that have been calling my counterpart in Rhode Island, state representatives and saying, hey, we need to do something about this. So the need from Rhode Island and the need from Maine sort of got us thinking that we should have a mobilization. So all the states in the Northeast sort of have this compact agreement where if there is there's a fire, if there's a forest health need, they will send staff from other states to help, to mobilize to help accomplish whatever task that might be. So this year it was winter moth, and we had people from our own staff from Maine, New Hampshire, Rhode Island, and Vermont. And we had a really good collection event. So we collected there's 4 collection events, but this particular mobilization was over 2 days. And we collected 11,267 caterpillars. And that includes some from Rhode Island. They did a couple of collections from Rhode Island. But yeah, about 7,700 of those came just exclusively from Maine. So you see this total number of caterpillars, 11,000. This is how many fly pupa that we got from that. So that's a pretty stark ratio there, and how much work it is counted, picking up these single caterpillars. And that's how many we have. We typically get so that's a total, sorry, 1,600, but not much different than 1,500.
Yep, why couldn't you just take the tarp and dump them into a bucket?
So there's a bunch of other stuff, there's predators, there's leaf material, and we also need to have a count of how many caterpillars we've collected, so that we know what the rate of parasitism is at each of these sites. So that we can sort of keep an eye on how well one site's doing versus the other. And if one site doesn't have a lot of parasitism, we will skip it and go to a different site. But yeah, so these caterpillars, once they're in the buckets, we send them down to some of them rear out themselves, but eventually all of them go down to our collaborators at UMass Amherst. They will finish rearing them out, and when they are in those little jerk pod pupa, they have a lot of grad students and undergrads that go through and dissect those pupa, open them up, and you can tell pretty quickly which ones have winter moth pupa in them and which ones have the fly pupa in them. And then they gather up all the fly pupa and then send them back to us with some statistical analysis. So, yeah, and so that 1,600 number, we have to split some of those with Rhode Island. So we kept about 1,000 and we sent the rest down to Rhode Island. So we had some sort of weird stuff going on this year. So typically it's just Maine and our staff that is doing these collections, and typically we do them over 3 or 4 days of pretty intense sampling. UMass folks come up and help us. So 1,600 is about what we get just with our main crew over those 3 or 4 days. So we had a lot more hands helping out with them this year, but unfortunately, the parasitism rates at a lot of those sites were lower. So even though we had more hands helping, the parasitism rates were lower, so it sort of evened out, unfortunately.
Just for the goof of it, if you were to double your take of caterpillars, do you have a downstream issue where they can't process them? Or if you doubled the volume that fed into UMass, could they handle that volume?
They're pretty good. They've been doing this for a long time, and I think they have the ability to hire like just undergrad students for like a work program.
Yes.
To make some extra money. So they don't really have a problem with the extra volume. Maybe if we like doubled it to like 30,000, it might be slower, but they're always really, really good, top-notch people down there. Okay, so these are all of the release sites that we have done in Maine since 2013, and some of these sites have gotten multiple releases. So Harpswell, since it was one of the first sites, has had multiple releases. Kittery at Fort McClary State Park, again, one of our first sites, has gotten multiple releases. And then South Bristol, Rutherford Island has gotten multiple releases. So we've been able to collect Cydenus from all of these previous release sites, with the exception of Vinyl Haven. We haven't been able to recapture Cydenus for some reason. It might just be sampling bias. This is what I was talking about before. It's the nature preserve that's about 5 miles from Fort McClary. And that's one of our best sites for Cydenus. So they do spread out, even if the initial release site sort of fades in its productiveness, the surrounding area almost always has a good population. So one of the other things that I didn't mention when I had the map up, so we have that infestation in Nova Scotia that's been here for well over 100 years, and then we have the mid-coast infestation down to southern Maine. So the lab at UMass Amherst, they really want to see where the intergrade is between where those two populations meet, because they are genetically different since they're from two separate introductions. So what we do is we collect at all of our sites, but then we go to Lubec to collect there. We collect at MDI, and we collect at a couple places in Down East Maine, just to see what winter moth population is doing, and then also, you know, where those caterpillars are coming from. So 2 years ago, and even last year, but 2 years ago was the first time we collected on Mount Desert Island, and you know, big winter moth population there, right near the ball field in Northwest Harbor. So we knew there was winter moth there, so we went to collect. We were not expecting to find any Sisenus there, but we did. It was very low number, but there was about 5 pupa that we got from Mount Desert Island. So the nearest release for Sisenus is a couple of hours down the coast. So how do those flies get there? And that sort of goes back to how well Sisenus, or how well winter moth moves with plant material. So a lot of big homes, a lot of wealthy homes, especially in Northwest Harbor, and a lot of landscape material moving, mostly in, but sometimes out. So what we sort of speculate happened is somebody either personally moved, you know, daylilies or lily of the valley or some plant from their yard that had soil attached to it that was in a winter moth infested zone, moved it up to their house or their property up there, and Sisenus came along for the ride, which is why it was there. So Sisenus can spread with winter moth, but again, just keeping in mind to not move soil material. But going more into the releases, so that's what we like to see. I hope there's no young viewers on there, but those are 2 flies making more flies. So that's what we like to see.
Bigger thing.
They're about the size of a housefly. Yeah, so everybody sees those cluster flies. They're just a little bit smaller than those cluster flies you usually see on your windowsill. So this is the emergence cage. So it's a wood frame and it's screened.
You can walk into a hotel.
So what we do, once we get those fly pupa back from UMass Amherst, it's sort of like a clandestine deal on the at the 7-Eleven in Kittery, because a lot of the time we can't get out of state approval. So UMass doesn't have those restrictions, so they drive all the way up and meet me at the 7-Eleven. And whoever's, you know, pumping gas at that point.
Be careful you don't get shot for.
Yeah, it's like, oh, look at that, look at that cooler full of stuff that's going on there. But anyway, we get those fly pupa, and then I will bring them to the next release site, and you can't see it, but in that box, there's a layer of sifted peat moss that's about that thick, and I sort of nestle those fly pupa in there, and then sort of tuck them in for the winter to have a nice long winter now. So a lot of times we do so they're typically going in right around this time of year, and then they're gonna be emerging the following April, typically late April, early May. And it's usually just before the red maple are starting to bloom. But what we do is we keep them in those, we keep them in that emergence cage, and we will peek in, just to see if they're okay, that they didn't drown. The signs on the top also help keep the rain from getting in there and drowning them. But we usually wait until we see mating. So with Sisenus, the females come out first, the males come out first, and they will wait around for the females in nature typically. So the females will come out, they need a little bit of time to mature, and then the males are there sort of waiting for them. But in the meantime, before the females emerge, they need something to eat because they're, you know, sitting around waiting, they need something to eat. So we'll typically get some apple slices, and we'll drip some sugar water on there, and we'll sort of put them in the corners of the cages so that they have something to eat, something to keep their energy up. It's still sort of cold a lot of the evenings and mornings. But once we observe that mating, then we know it's release time, and that's typically when we see that mating. It's around the time that the red maples are blooming, and so after the female mates, again, she has to mature those eggs for a little bit. So both males and females will go to those red maple blossoms and feed, as well as other flowers. And then I'm not quite sure what happens to the males. I'm pretty sure that they have done their job and they pass away. But those females, remember that word from before, cenobiogenic, they will keep laying eggs for their whole life pretty much. And they'll just keep maturing eggs and laying them. So with the females, are doing once those eggs are mature is that they are sort of sniffing out where that leaf damage is happening. And it's a combination of host volatiles, so those trees are giving off these volatile chemicals because the leaves are damaged. It's sort of like when you cut grass, same sort of idea. And then so the female flies are smelling for that. So when they go to that leaf damage, there's also other cues, and I believe that they can also pick up the saliva from the caterpillars feeding, because they do produce saliva. And they're able to sniff that out, and then they start laying their eggs. Those really small microtype eggs, about the thickness of a sheet of paper. They lay them. Caterpillars are still feeding on those leaves, so they ingest them, and then the egg sort of lays dormant in them until the caterpillars in late May, they're done feeding from the trees, they drop off from the trees, bury into the soil, pupate, they spin that cocoon, pupate, and that's when the egg hatches in them, and that maggot will eat them from the inside out. And then the following spring, it is a fly emerging from that pupa.
Sorry, but this is great, fantastic. Can you give some numbers here? I mean, I'm thinking you got this one little Cyzenus fly lands on my oak tree, and there's 10,000 of these crazy winter moths on there. What's the ratio of Cyzenus to winter moth that's effective? You know what I'm saying?
I don't have an exact number
No,
for
of
you,
course
but
not, but yeah.
so a lot of these releases, especially the earlier releases, those were like 2,000 flies a piece, and our recent releases have been about 1,500 flies, and we've done
That's per-
like 500.
My apologies, so just from my perspective, so would Bath get 2,000 flies? Or did this whole list get 2,000 flies?
It depends on
Yeah.
where they were.
Yeah.
In the earlier ones got the 2,000 because
Okay.
they had a lot from southern New England
Yeah,
that
sure.
UMass was shipping out. And then later, as we couldn't rely on southern New England and are just our own collection efforts, those got 1,500. So South Bristol, they got 2 releases. That first release was only 500 flies because we had a really bad year. So in order to boost those numbers, we had to do a second release there. So when we do those 1,500 or 2,000 releases, within a year or two, you start seeing noticeable reduction in defoliation. So obviously all of those flies are not female. It's like a 50-50 ratio, but each one of those females, again, I don't have an exact number, but it's like on the order of 1,000 or thousands of eggs that each female lays. So you can have a pretty big population reduction in a short amount of time relative to how biocontrol works. So there's other biocontrols. So there's a wasp called Agripon, and that was released in Nova Scotia in the 1930s. Was not released here in the US just because there was doubts about how specific it was and if it attacked other species. But if Canada releases it, or if we release it, there's no, they don't obey the border. So we've been sort of keeping our eye out to see if any our collections. And we did pick up Agripon in one of our southern Maine sites, but there's also 40 different species. So UMass Amherst was trying to go through and do some genetic sequencing to see what species it belonged to. But yeah, like I said, very likely to travel with winter moth in order to and spread with it. So I wouldn't be surprised if it does show up here, even though it's been here for about 100 years in North America. One other important thing that a lot of the research that lab down at UMass has done is soil predators are very important in controlling winter moth numbers. So that's a ground beetle, but there's centipedes and all manner of soil arthropods that will prey on the pupa when they're in the ground. And they don't distinguish between winter moth pupa or the Cyzenus, so sometimes they can be detriment. But the study that they did show a lot of mortality just from the native natural soil growing arthropods. Almost getting close here. So we also do a monitoring for winter moth. Again, it's only the males that are flying, but the males do give a good indication of what the population is doing. So earlier on, we did have a more robust monitoring program. We used to hang about 70 traps from Kittery all the way up to Ellsworth, Bar Harbor, and then in later years we would go down east and to Lubec. But there was a problem with basically there's two manufacturers of the lures here in the US, or US and Canada, and they stopped making the lure. So there is a company, the lures used to be about $2 a piece. The lures are now $10 a piece, and that was a couple of years ago. I haven't looked to see what they are now. So with budget constraints, we can't trap and monitor, we can't run 70 traps, unfortunately. So we are relying on a lot of the work that UMass Amherst is doing. And each year it's sort of a different goal, but what they're helping us monitor by running some of their research programs up here in Maine. And other states in the Northeast are also in cooperation with those folks at UMass. So this thing that looks like an eraser, it's a rubber septum that's impregnated with the pheromone that these female moths that are flightless, they release. So that's hung right up in this little basket here. So the male moths smell that, they think it's a female moth. They try to get in there and they collide with that and they fall down a funnel. There's a little Vapo kill strip in the bottom, so that so that captures them and sort of preserves them, so that when we come back at the end of the season, or if it's a weekly check here, we'll collect the contents of that and keep it hung up basically until like the middle of January, until we know that there's no more winter moths flying.
But that's insufficient. For example, this summer, I used an equivalent for Japanese beetles.
Oh, yeah.
And that thing would fill up every day. It was amazing. But there's no equivalent to that kind of a trap and kill kind of pheromone based thing for winter moth, it sounds like.
Unfortunately not. So biologically, unfortunately us males don't matter too much biologically. It's the females that really keep the population propped up, so it only takes, a smaller amount of males to keep that population built up. So also the thing with Japanese beetle traps, if you don't like your neighbor, you're gonna want to make sure that those traps are in their yard, because what you're doing is you're drawing all those Japanese beetles from the neighborhood to your yard.
My hood.
And it attracts males and females, so it's a little bit more effective, but it sort of pulls them in from a little further.
Where does the mating occur, on the ground?
It can occur on the ground, but most often it occurs on the tree trunk. So when those females emerge from the ground, again, they can't fly, so they have to crawl up the tree trunk. And then once they reach a spot where they're feeling like they want to meet Mr. Right, they release some of that pheromone and call the males to them and mate with them there.
What if we just sprayed insecticide on the base of our trees? Does that do that?
There's a few different things. There's some non-target effects, less because winter moth is active during a time where a lot of our native insects aren't. But a lot of those insecticides can't be used under a certain temperature, or some of those formulations can't be used under a certain temperature. But it's not a recommended management strategy.
Because they're mating right now, give or take, or a few weeks.
Yeah, they'll be emerging within the next, it's the 20th now, so it'll probably be three more weeks, something like that, so you start seeing moths.
So do you guys dissuade us from using insecticide? I would say if you spray for ticks around your house,
Yeah,
same stuff would kill a moth, right?
it would, but it's not.
Also kills this siziness, the other fly, right?
Well, not if you're applying it in the winter, but I'll get into some of the recommended management strategies, but I would say that spraying, like doing a basal drench of your trees in the winter is not not recommended. One, because it's depending on the temperature, the tree bark will absorb that basal spray, because a lot of those sprays are designed to be able to be taken up by the tree and not be like perpetually wet, so that animals or kids or whatever come in contact with it. So a lot of that will be absorbed and not be as effective. But then there's also the temperature, again, it's hovering around freezing around that time. But there's other strategies that are more effective, which is my next one. Okay, so there's a few different options. So I should note that none of these are gonna solve the winter moth problems on the island, on some of these peninsulas. It'll basically just preserve individual trees that you don't wanna see defoliated. There's no landscape-wide management for winter moth other than that fly site beds. So during late fall, early winter, again, when it's above freezing, so when you have a lot of these insecticides, that label that comes with it, so that label is the law. So if you apply, twice the recommended amount, if you apply not in the way that pesticide is formulated, it's a violation of the pesticide laws that are federal, but that we have here in Maine. And that's just because a lot of these pesticides, they go through rigorous testing, and they're engineered to do a certain job at a certain dosage at a certain time and a temperature. So all of that's already been figured out, and it's all in that instruction or the label. But anyway, so late fall, early winter. So once those eggs are laid, you can use a dormant oil, which is they're also called horticultural oils. You can spray those on, and they stick to the bark and they stick to the eggs and suffocate the eggs so that the caterpillars will not hatch out the following spring. But again, they can't be applied under a certain temperature, or they don't work for as per the label. Yeah, so also when the eggs are newly hatched, so that shouldn't say late March, it should say early April. There's a spray called it's called BT, and BT is a soil bacterium that produces an enzyme that erodes the gut of specifically caterpillar and moth larvae, which are the caterpillars, but it doesn't affect any other insect groups. It's just Lepidoptera, which is that group that encompasses butterflies and moths. So BT is effective on very young caterpillars. What happens is when you spray it on the foliage, those caterpillars eat it, those young caterpillars eat it, and it erodes their gut so that they can't keep feeding, and then they eventually either get an infection or die from lack of nutrition. But again, very specific to caterpillars, and it also breaks down in the environment very quickly. It breaks down in sunlight. So that's one option that's safe for if you're worried about non-target effects. And then this chemical here, spinosad, that's a general insecticide, so that will kill any insect that it gets on. So that is something to be used with more caution. And I should also note that with these two sprays in the middle there, if there is an area that's near a Cyvenus release site, you also risk killing the Cyvenus that are inside those caterpillars. So if you're close to a release site, we typically recommend that you don't spray, but it's sort of a tool in the toolbox. If you have a specific tree or a blueberry bush or whatever that you don't want foliated, that's an option. But again, really tall trees, you're not going to be out there spraying with a pump sprayer. So a lot of these will need to be applied by a licensed pesticide applicator or a arborist that's licensed pesticide applicator. But probably the reason why we're having this in mid-October is so that you guys have this information for the spring or for the coming weeks here, where we're going to start seeing winter moth come out. So, and I refer specifically to these tree bands. So it's these sticky tree bands that get wrapped around the trunk of the tree, and it has a glue, typically that glue is called tanglefoot, it was used for orchardists for birds back in the day, but there's other more friendly uses for it now, well, friendly for our avian friends. So basically, you wrap these bands around the tree, and the idea is that when these female moths are crawling up the trunk of the tree, they will get tangled in that glue and not be able to lay eggs and die. So one of the important things for those tree bands is basically to, before you put that tree band on, is to get some cotton batting or some foam soft foam that will conform to the bark of the tree, because you don't want these females basically crawling underneath the band and just bypassing it. You can buy them pre-made. A lot of the, I believe it's O'Donnell's, no, sorry, I'm thinking that's other Maine.
Hardware store.
Yeah, the
Hardware.
hardware stores, but then also some of the local garden centers, will have these bands because it has been a little bit prevalent in this area of Maine. So you can buy the pre-made stuff. I've seen people just get the glue, which you can buy in a can, and use saran wrap around the tree, and that's just as effective. As long as it's all crumpled up, but you can buy them pre-made or you can make them yourself. And in areas that have a really high infestation, you're going to want to put 2 bands on the tree, because sometimes those bands can get really saturated with females or with the males, hormones that the females are releasing. So those can get saturated, and if those get saturated, then they can just sort of walk, unfortunately, over their comrades up the tree.
How high up the tree?
Just head height or below, just as long as you wouldn't want to put it like down by the root collar, you just want to put it above where there's sort of like a nice uniform surface, so you don't have to worry about filling all those gaps underneath.
How about duct tape? I read something.
Maybe less effective, it might catch a few, but the glue, even on something like duct tape, if it gets dust on it, it's not as powerful of a glue as something like Tanglefoot or the glue that a lot of these sticky bands come with.
I mean, if you put duct tape around and then put the Tanglefoot on top of it, on top of the duct tape.
Yeah, that could work. You would probably have to, you sort of want the width of the bands to be at least that wide.
Oh, okay.
Something like that would just get really saturated really quick.
I'm sure tape's too expensive.
Sorry, so again, just reiterating, winter moth moves with landscape material. So basically, anytime from early June all the way through November, there's gonna be pupa in the soil. So, digging up hostas and gifting them to a friend, or one of the ways that winter moth came to Maine, a lot of people have second homes, but their primary homes are deep in the infested area down in southern New England, and they unknowingly brought some of these, up to camp or up to their house. So just be aware of that. So that's like plant sales and gifting plants and again, moving plants from one property to another. And they're very, like I said, they're like a tic tac. They're very small, very almost indistinguishable from the soil around them.
How deep do they go?
I think they can go down like 5, 6, 7 inches, something like that, but it's definitely not a foot, but within that top layer like that. I think that's all I have for you, but I'm here to answer any questions you have. I'm glad that nobody threw tomatoes. Yeah.
No.
Do you guys have any concern with hybridization with the Bruce bandworm and then the fly not being able to? You said the fly was very specific to work to the winter moth.
So there is some hybridization, because even though Bruce bandworm's active a little bit earlier in the year, there is some overlap between winter moth, and again, they're very closely related. And so the folks down at UMass Amherst, that's one of the things that they do with those traps that we trap the males for. They sort of do a genetic sequence in all the moths in that trap, And there's they've been giving us a percentage of, Bruce bandworm, winter moth, and then hybrids, and the hybrids do occur, but it's a much lower, rate than what you might think. But I don't have any specific info about Cydenus, on the hybrids, unfortunately. Sorry, there's.
Have you noticed that the outbreaks of the winter moths are cyclical like the brown tails?
They're a little bit cyclical, so I will say that even in Kittery, which is one of our oldest release sites, and one of the earliest infestations in Maine, we do still see outbreaks there, so it's not that Cyzenus prevents the outbreaks at all, but it makes the outbreaks less severe and it makes them last a little bit less, but it's just part of winter moth's biology, to escape its predators every now and then, and a lot of that has to do with, environmental conditions the previous year or the current year, and what Cyzenus is doing, if it has a high success rate or not. So we'll note that, again, even in its native range, winter moth sort of fulfills a really important role, and our native Bruce bandworm also here fulfills a really important role. So, have you guys ever heard of climax forest? So it's for primary, secondary succession. So basically, in the Northeast, the climax forest is basically what all of this forest would turn into if nothing was done to it, no disturbance, no fire, no hurricanes, no humans. So the climax forest in most parts of the Northeast is beech, oak, hemlock, maple, that sort of stuff. So in that climax forest, you just have those 4, 5, 6 different species. It's not a super diverse forest. So what happens is that, so Bruce bandworm here will defoliate the oaks or the sugar maples, and then stuff like cherry or birch or other species that are growing in the understory. They have light for a month or so, and they grow up a little bit, but then the oaks and maples releaf, and they sort of stop growing because they're shaded. And then that happens again in another 10 years, and then they grow a little bit more. And then after that happens, over 3-4 decades, those trees are able to be co-dominant with all of those beech and hemlock and oak and maple. So a lot of people call insect outbreaks a slow-moving forest fire. Just like we have learned that forest fires do play a critical role in the health of our ecosystem by creating that disturbance and creating that that biodiversity. These insects do the same thing, and winter moth does a similar thing there. So getting back to that outbreak cycle, they do outbreak, and it's just part of biology because they're fulfilling their higher power, if you want to say, and it just comes out the total of people's trees sometimes.
Has there been any thought of having like a community gather the winter moth caterpillar?
We have
At an event?
We have thought about it. So even just that deployment, and that was 15 people, again, a mix of our own staff plus Rhode Island, New Hampshire, Vermont. That was a lot. Even just 15 people is a lot because you're sort of, it's like a gamble and a juggle. So when we were collecting those caterpillars, we're trying to collect them when they are the most mature so that we don't have to feed them branches for weeks on end. And the longer you have them in the buckets rearing them, the more die because they're so crowded and packed together. So you want to get them when they're the most ripe, but you also run into the problem that you might miss them if you wait even a few days. So it's very stressful time. Late May, early June was very stressful for me. This year was actually stressful, one, because we had more release sites to collect from, which is a good thing. But also, there's a different, as you know, being down here on the island and in the peninsulas, there's a different microclimate than even up in like that. So the host phenology and the caterpillar phenology was way different down on the peninsulas than it was up in Bath. So just for instance, the oak trees were fully leafed out in mid to late May in Bath and the mainland. Down here, I'm sure you noticed, everything was about 2 weeks behind. So that means the caterpillars are 2 weeks behind, but you only have this one collection event, or you're trying to coordinate all these people coming from out of state and hotel reservations and all that. So we ended up doing like 3 or 4 different days just with us, but then also with these collaborators. So it's a lot to juggle, but I'm sure if we maybe put our mind to it a little bit more, and it's not like the communities need hotel rooms because they have their own hotel rooms there. So it is something definitely to think about. But it was a little stressful. I'm still kind of recovering from it this year. But yeah.
The controls that are not the fly, that are specific to caterpillars, they're not specific to bed caterpillars. They also look at, the monarch.
Yeah, you're talking about like BT and stuff, or BT.
exactly.
Yeah, so that's also something to take into consideration. So oaks in particular host many native species of caterpillars, both moths and butterflies. One of the good things about the biology of winter moth is that it's one of the first caterpillars to hatch out in the spring, and it usually hatches out, way before a lot of our other native species are out. So that sort of decreases the risk for non-target effects. But if you apply BT later, like in early or mid-May, then there's a lot more of our natives that are out. So it's a consideration. So the Maine Board of Pesticide Control, which is part of the Department of Agriculture, Conservation and Forestry, which we're also part of, their motto on their logo is think first, spray last. So thinking about all these non-target effects. And one of the reasons why Maine has very strict pesticide rules, even sometimes stricter than EPA. One of the major reasons is to protect the shellfish industry on the coast. Because not the ones that are listed, well, I think maybe spinosas, but some of these are highly toxic to shellfish. So obviously we want to prevent that's why there's setbacks from the water. There's certain things that you can do within the 0 to 50, 50 to 150, and then 150, 250 and beyond. There's only certain things and certain products you can do within each of those boundaries, and that's to protect the lobster and the shellfish industry.
Are there any plans to release the fly on Westport?
I knew that question was coming. There is, it's kind of again, that's kind of like a juggling thing, since we are really limited in the amount of biocontrol that we have. It's not necessarily who screams the loudest. It's more based on, we definitely take that into consideration, and that's the reason why I'm here. I had a lot of calls from Westport Island. But there's also, we have to rely on aerial survey data and stuff like that. And if any of you were down on Georgetown, Pittsburgh, that was also really horrible. And Bristol continues to be horrible, but they have those 2 released there. So they're sort of dropping back on the list, and we would prioritize something like Westport Island or Georgetown, Pittsburgh area. But yeah.
It seems like there must be a critical mass of Suzennas that you can use. You can't put 50 in each town. It sounds like you have to have some volume to keep them reproducing. Yeah.
It worked, yeah. So these
like each other.
These protocols that were developed by UMass Amherst and us, and then also the folks out in Oregon from the 1950s. There is sort of a sweet spot. If you don't release enough, say if you were to split that all into 50s, it's a big world and they're small flies. So yes, they can meet up at the local bar, the red maple flowers, but there's less of a chance of them getting together. And that's one of the other reasons that even if we're releasing 1,500 or 2,000, we keep them in that cage until we see that mating, so that we know at least a certain percentage of those females mated, they're gonna be producing eggs. Cause you wouldn't wanna just open it up and whatever comes out without having that sort of singles bar in the ground before we open it up. But the sweet spot, like, ideal would be 2,000 per site, 1,500 is still excellent. And then when you get below 1,000, it's sort of less, it's less worthwhile. It's still worthwhile to get some flies out there because they do continue to lay eggs throughout their life cycle, but it's less productive. So that's the reason why we did 2 releases in Bristol, because the first release was only, I think it was like 543 flies. So we needed to do another release there just to boost the numbers and move up that peninsula and over to the other peninsulas. So, yeah, to get back to your question, yes, you guys are definitely on the list, but it's.
And when you release them, do you like sort of open a box up here right at the town office? You do it every mile all the way down the main road, or how are they released?
So that emergence cage that we bury in the ground right around this time of the year with the pupa in it. So it's just a single release at one site, and typically what we look for is, again, a lot of oaks, a lot of red maple, because those are hosts for winter moth, and we want winter moth to be close by for Sizenis. But then we're also looking for areas that have like contiguous forests. So like the South Portland release site, that was at a homeowner's residence, but that also backed up to a lot of parkland. So it was sort of the best of both worlds. We would be able to have those homeowners there to monitor the flies, so we didn't have to drive down to South Portland every day to check, to see if there's mating. But then also, you had all that parkland. So the past few releases have been on like land trust land or like nature preserve land, stuff that we know is not going to be sprayed. That's also another criteria. You don't want to be treating the trees that you release the Sizenis at or nearby. So we know those preserves aren't going to be treated. We know they're not going to be cut down, hopefully. And then we know that there's lots of food for them, for the flies, both the larvae and the adults that need that nectar.
Yes, as bad as the outbreak of larvae we had this spring, the oaks and the maple, or the oaks and the apple around our house anyway, they seem to have a second wind, and they foliated pretty well. Is that typical, or does the outbreak get to a point where there's just so many that they continue feeding and the foliage doesn't recover?
No, so a tree, and I'm sure many of you have seen it, a tree can be totally stripped. So oaks and maples in particular, that's part of their evolutionary strategy. A lot of caterpillars eat oak, a lot of caterpillars eat maple. So they're kind of betting on that. They have reserves, and once the feeding is done in late May, early June, and they don't sense that feeding pressure anymore, they will put out a new set of leaves. So it's very common. What complicates things is, again, like other defoliators like browntail, they stay caterpillars until late June, so they're going to keep feeding. So additional defoliators on those trees, but then also stuff like drought, like the one that we're having this year, and especially on the islands and the peninsulas, there's a low depth to bedrock, so a lot of the soils don't hold as much moisture as deeper soils on the mainland. So there's less moisture available, and you can imagine how much moisture it needs to be able to push out whole canopy of leaves. I will say that basically, if you had to pick between browntail and winter moth is the way to go. One, because unless it's very severe, they don't consume the entire leaves. It's just the shottle pattern. Those leaves can continue to photosynthesize, provide food for the tree. Birds eat winter moth. Many birds don't eat browntail because it's irritating and hairy. And then also, they don't give you a rash, like ractopus. But yeah, so. I know you guys had Ractail a little while ago, and winter moth is severe down here, but. And also, we might see some mortality from some of these trees next year, especially the ones that sort of fail to put out another set of leaves. Because of that added stress of the drought, but. 90-something% of the trees will be just fine, they'll push out another flush of leaves. Some of them might have some branch dieback of some of the smaller branches, but that's very typical of any defoliation. It just takes a lot of energy to push out the.
And we don't have any acorns this year.
Yeah, so that's another thing, is the caterpillars, again, in mid-April.
No acorns, no apples in my.
They'll eat any tender foliage that they can, and that includes a lot of catkins, that, develop into acorns. So, yeah, it's a mass, in some areas it's a mass year for oaks, but, yeah, not in some of the really denuded areas, unfortunately. But the squirrel will be fine,
Yeah, I was gonna say squirrel.
put out extra feeders.
So do you, I'm sorry,
Go ahead.
do you anticipate then in the spring, in this coming spring, that it'll be pretty bad down here?
It's sort of hard to say, so.
Worse than last year?
I would say that it would probably be about the same, unless, I didn't mention them, but there are pathogens that are associated with winter moth. It doesn't play as big of a role as it does with brown-tailed moth. So brown-tailed moth is a fungus and a virus. There are other pathogens that attack winter moth, and we do see, like when we're beating onto the tarps, usually we'll see some dead caterpillars, and in some sites it's a lot, and they're kind of like black and necrotic. So unless there's some stuff like behind the scenes that we don't really directly observe a lot of time, just because it's in the canopy, but those do have an effect on the winter moth population. So I would expect it to be about the same, but again, Zizanus is on its way, if not already here in some small measure, but then also a lot of those pathogens. So it's kind of like with humans, if it's getting colder, we're all packed in. And so the higher the winter moth population, the closer contact those are, and that's sort of right for pathogens to sort of spread through. We haven't seen any, major collapses directly attributed to the pathogens, but they are around and they do kill some caterpillars. But yeah, I would expect the same and hope for better.
So in the spring, when we're walking and we pick off 20, because if they're ballooning, they're like ripe and fat, right?
Oh, yeah.
Okay, so, no, do you want me to collect them and bring them to you?
Maybe, there's the tomatoes that we were talking about.
A little dead stuff.
It would, one of the things that would be interesting to actually do some collections in West Rhode Island, just to see if Zizanus is here, because it does show up sometimes when you don't expect it, but I would expect it to be here at some smaller level, just because on either side of you, there has been multiple releases. So Booth Bay, East Booth Bay, both of those, each of those got a release. West Bath got a release, like that whole west. So it's sort of all around, and it would really surprise me if Zizanus wasn't already here. But sometimes it just takes a little while, and like I was mentioning, we could certainly do a release here. I just have to. We're having a problem with the aerial survey data this year. Just it's a new mapping program. But once I have that done and tabulate how many calls came from each town, I should be able to do a release down here.
Can we all call?
I can.
Yeah. Well, I met Ann's niece at the conference that I was at yesterday and earlier today, and I mentioned, I was like, I don't know if they're like organizing or something, but like I get so many calls from Westport Island, and she was like, yeah, they're probably organizing.
Oh, shoot.
All the places.
Is they organize.
She talked about it.
Flies in her refrigerator.
Oh,
Jordan, do you say?
yeah, I don't know if you're supposed to say that at all.
It's, if there was anybody else, I would be concerned, but it is on the
It
fourth
was
step
something,
down there.
yeah, we were on the phone. Anyway. I don't have them, I don't have.
So if you were gonna do a release, that would be this fall, so that they would be ready.
Yeah, so it would either be this fall or next fall. Well, so we would put the cage in the ground in the fall and then the releases are in
Early spring.
yeah, like early May.
We want one this fall.
Yeah. That's great. Here to gauge the level of enthusiasm.
Keep on soon.
Very enthusiastic.
Well, and if you ever need volunteers, you have my email, cause I'm sure, there's just so much concern about it, you know?
Yeah, definitely. And like, it's wonderful to see a community engaged like this because most of the time when I do community talks or if there's community engagement, it's with brown tail. And usually it's just a small subset of people that, you know, care about it. But it's nice to see a good turnout and all the folks on Zoom. But it's good to see that community engagement.
I have one more question.
Yep.
Why are the winter moths, along the coast and not further?
So that was like the $1 million question. So again, the folks down at UMass Amherst, they have been doing a lot of research. So one of the current theories is that, again, we have that microclimate along the coast. And further inland, it's a little bit harsher in the winter. So when those adult moths are emerging from the ground, a lot of that is dependent on the soil temperature. So it needs to be warm enough for them to emerge. But if we have a really early freeze where the ground sort of stays frozen for a little while, that negatively impacts winter moths because the adults can't emerge, they can't break through that frozen ground, and then they just die underground. But being close to the ocean, it's a little bit more mild, a little bit more temperate, so that they're able to punch up through without that freeze.
Thank you.